What if an amputee could regrow their limbs? Well, what if we told you that there are animals that have this ability? One of these is Asterias rostoni, a member of clade Asteroidea. Starfish, also known as sea stars, are part of the kingdom Animalia, phylum Echinodermata, and are members of class Asteroidea. Many individuals are familiar with starfish due to their unique shape, and the fact that they are able to regenerate parts of their bodies, such as their limbs, which sometimes get damaged.
As invertebrates, sea stars are extremely good at limb regeneration, a process that is limited in vertebrates. With this ability, they become one ideal model in the research of echinoderm regeneration as a whole. If needed, they can even regenerate all 5 arms at the same time. However, this process would take many months to accomplish. One research paper, Patterns in Cellular Mechanisms of Arm Regeneration in Adult Starfish Asterias rostoni Bell, investigated and analyzed the mechanisms in which starfish regenerate certain cells to form a new limb. This research paper was the first to study this strange phenomenon. In this study, starfish between 10 and 15 centimeters in diameter were taken from ocean waters and placed in tanks. In the experimental group, the arm tips of each starfish were amputated and removed, while the control group had their arm tips amputated and essentially glued back on after 2 days.
Scientists found that after 4 days, the amputated arm stumps began to regenerate, starting with the epidermis. Pre-epidermis, with 2 to 4 layers of round cells, were formed on the outside surface of the wound. These cells are undifferentiated, or cells that give rise to other cells, similar to stem cells. After only 12 days, new tips were forming, and after 53 days, the starfish had regenerated the full length of its arms, which can be seen in Figure 1, C and D. A and B of the Figure 1 show where the tip was amputated and the arm length over 60 days.
The cell dedifferentiation, when cells regress from a specific function to a simpler state, and proliferation and increase in the number of cells involved in epidermis formation, is different from the wound epidermis formation of limb stumps found in adult newts, which is achieved strictly through cell movement without proliferation. This indicates that the invertebrate wound-induced epidermis formation is different from that in newts.
The starfish in this study first went through a period of the dedifferentiated cells traveling to the wounded sites of the arms. They then formed a thickened pre-epidermis that matured and formed the outer epidermis and inner parietal peritoneum, which lines the cell walls, which formed blastema-like structures with differentiated dermal cells. These blastema-like structures can be seen in mammals, amphibians, and annelids. Through this complex process, starfish achieved arm bud formation, as well as arm stump elongation, at the same time, which explains their incredible regeneration abilities.
Because of the striking regeneration potential and remarkable ability to regenerate lost body parts and injured organs, along with the close evolutionary relationship and developmental similarities between echinoderms and vertebrates, sea stars are a valuable experimental model for studying all aspects of regenerative processes from molecular to macroscopic levels. The research done by this study is an integral part of the study of epimorphosis, the process in which animals regenerate parts of their body. The results of this study are the first of its kind, as little is known about the patterns that occur in starfish arm regeneration and are applicable to echinoderm epimorphosis as a whole. From the findings, researchers now understand that there is much to learn about how epimorphosis occurs depending on the severity of an injury, the environment a starfish is in, and how different species of starfish undergo epimorphosis.